Okay, so welcome to this next video in which we are discussing the soluble guanylate cyclase enzyme. Okay, so this enzyme is very important in the endothelium-derived relaxation factor pathway because the nitric oxide, which the endothelial cells are releasing, goes up, diffuses to the smooth muscle cells, and in the smooth muscle cells it's going to activate these soluble guanylate cyclase enzymes. Okay, so I now want to discuss the fact that there are two different soluble guanylate cyclase enzymes. Okay, so there is one known as the alpha-1 beta-1 soluble guanylate cyclase, and this, as you might imagine, has a specific alpha subunit. Its alpha subunit is alpha-1. So this half here is the alpha-1 subunit, there. And in place of its beta subunit is basically this beta-1 subunit. Okay, so this one is beta-1, right? Okay, and it's the one which is found in the vascular smooth muscle. So you find this alpha-1 beta-1 soluble guanylate cyclase in the vascular smooth muscle. And what's significant about it is that it is not membrane bound. It's within the cytoplasm. It really is a soluble guanylate cyclase. Okay, now let's have a look at another one. Alpha-2 beta-1. So again, you still have this beta-1 subunit as your beta subunit in the soluble guanylate cyclase, but now, instead of having an alpha-1 subunit as your alpha subunit, instead you have this alpha-2 subunit as your alpha subunit. So this is your alpha subunit, alpha-2, and your beta subunit is the beta-1 type, basically. Okay, now this type of soluble guanylate cyclase isn't actually within the cytoplasm. This is found in neurons. Okay, and basically it is it's membrane bound. The alpha-2 subunit of the guanylate cyclase, it basically is able to interact with some protein in the membrane of cells. So there is a protein in the membrane of cells, and here, let's say, is the alpha-2 subunit interacting with it. So I'll just draw the enzyme as two sort of rectangles like this. Let's say this is the alpha-2 subunit, this is the beta-1 subunit, so let me colour things in. So in pink here, this is our guanylate cyclase enzyme. Okay, the two subunits of the guanylate cyclase enzyme. And I've got the alpha-2 subunit here, so it's this alpha-2 beta-1 soluble guanylate cyclase, and it's interacting basically with some membrane-bound protein here in this turquoise colour. Right. Now, okay, so this means that this soluble guanylate cyclase is localized at the cell membranes of cells, and it's significant in neurons because it's believed that this helps you target the soluble guanylate cyclase to synapses, basically. Let me just go over a little bit of theory concerning neurons. Okay, so in neurons there is this phenomenon known as long-term potentiation, which is basically if, let's say this is the presynaptic neuron here, so this is presynaptic, and this is the postsynaptic neuron. So this is presynaptic, and let's say this is the postsynaptic neuron here. So this neuron here can release glutamate, let's say, onto this postsynaptic neuron. Glutamate is the main excitatory neurotransmitter in the brain. Okay? Right. So, when this presynaptic neuron releases glutamate onto this postsynaptic neuron, it will cause a slight depolarization of this postsynaptic neuron's membrane. So it will cause an excitatory postsynaptic potential. Okay? Now, if that excitatory postsynaptic potential in this postsynaptic cell is coupled to this neuron actually undergoing an action potential, i.e., if this presynaptic neuron stimulates the postsynaptic neuron when the postsynaptic neuron happens to undergo an action potential, then what will happen is this synapse will get strengthened, i.e. the synaptic communication between these two will strengthen. And what do I mean by that? I mean that if you stimulate this presynaptic neuron to release neurotransmitter onto this postsynaptic neuron, the excitatory postsynaptic potential it will produce in the postsynaptic neuron will now be greater. That's what I mean by long-term potentiation. Okay? So the effect of stimulating the presynaptic neuron on the electrical potential difference across the membrane of the postsynaptic neuron will be greater. Okay, now, how does this phenomenon occur? Well, there are two arguments, basically. One could be that it's a presynaptic modification. What you could do, basically, is you could basically fill this presynaptic vesicle with more glutamate vesicles, so that every time you stimulate this presynaptic neuron, you're going to release more glutamate into the synaptic cleft, basically. That's one suggestion for how you could increase the effect of stimulating the presynaptic neuron on the postsynaptic neuron. Another suggestion is that it could be postsynaptic, i.e. you could put more receptors for the glutamate in the postsynaptic membrane. That would also strengthen the effect of the presynaptic cell on the postsynaptic cell. Now, there are arguments in favor of both, but in the case, if it is the case that presynaptic changes happen, then what would have to happen is the postsynaptic neuron would have to send some sort of message back to the presynaptic neuron to tell it to increase the amount of glutamate vesicles in this presynaptic nerve terminal. And that messenger has been suggested to be nitric oxide. Okay, right. That's why people think that this may well be important in neurons, because they think this alpha-2 beta-1 soluble guanylate cyclase may well be localized in these presynaptic nerve terminals here, basically, and that may then carry the signal from the nitric oxide, which is this retrograde messenger. Okay, so this is hypothesized to be the retrograde messenger, although this is still controversial science. It's not agreed upon. Retrograde messenger. Right. So we'll continue this video, and then we'll continue this discussion in the next video.